Translation involves decoding a messenger RNA, mRNA, and using its information to build a polypeptide or chain of amino acids. For most purposes, a polypeptide is basically just a protein, with the technical difference being that some larger proteins are made up of several polypeptide chains. So similar to transcription, the process of translation also takes place in 3 key steps: initiation, elongation, and termination. In contrast to transcription, though, the process of translating mRNA into proteins takes place within ribosomes, which, if you remember from our previous discussion, ribosomes can either be found free-floating in the cytoplasm or attached to the rough endoplasmic reticulum. Now, to understand these 3 steps of translation, initiation, elongation, and termination, we must first break down the definition of a new term, codons. Codons are basically nucleotide triplets. That is, they are a sequence of 3 nucleotides, which together form a unit of genetic code in a DNA or RNA molecule. Ribosomes read mRNA 1 codon at a time, and each codon codes for 1 amino acid. So, the order of the codons in an mRNA sequence determines the order of amino acids in a polypeptide chain. This is where the decoding happens and why this process is called translating, because it's translating the genetic code in codons into amino acids, which are then bound together one at a time to form that polypeptide. So, to ensure that the whole sequence of mRNA is translated into a protein, meaning we are making sure that translation is not somehow starting in the middle of the mRNA sequence, the ribosome reads for a quote-unquote start codon. A start codon is the sequence AUG, found at right after the 5' cap, at the beginning of all mRNAs that are ready for translation. Initiation is when we have a ribosome divided into small and large subunits, an mRNA sequence, and an initiator tRNA that is carrying the amino acid methionine, which corresponds with that AUG codon. When we have all of these components coming together to form that initiation complex, that is when we have initiation of this translation process. So the initiation complex is the molecular setup needed to start making a new protein made up of all the units I just described. So to break this down, first the tRNA carrying methionine attaches to the small ribosomal subunit. This tRNA also has an anticodon, UAC, associated with it, which just so happens to be complementary to AUG, right? U associates with A is complementary to U, and C is complementary to G, which is our start codon. So this is how initiation takes place in translation. Together, the ribosome binds to the 5' end of the mRNA by recognizing the 5' cap. Then it quote-unquote walks along the mRNA in the 3' direction, stopping when it reaches the start AUG codon. The tRNA's UAC anticodon then binds to the AUG start codon, and the second step of translation is ready to take place: elongation. Within the ribosomes are 3 tRNA binding sites, labeled A, P, and E. During the elongation step of translation, tRNAs enter the acceptor site, or the A site, and the anticodon on the base of the tRNA pairs with the codon in the mRNA, complementary pairing. A peptide bond then forms between the amino acid from the A site with the amino acid from the P site. The P in P site technically stands for peptidyl tRNA binding site, but I like to think of it as the peptide bond site, because this is where the tRNA goes after its amino acid forms a peptide bond with the previous amino acid. The formation of each peptide bond is catalyzed by an enzyme called peptidyl transferase. After the amino acid in the A site forms that peptide bond with the growing polypeptide chain, meaning with the amino acid adjacent to it, the ribosome will move 3 nucleotides, so 1 codon, towards the 3' end of the mRNA. This movement transfers the tRNA with the growing polypeptide to the P site and allows the quote-unquote empty tRNA to then exit at the E, or exit site, of the ribosome. During translation elongation, the mRNA template provides tRNA binding specificity. As the ribosome moves along the mRNA, each mRNA codon comes into register, and specific binding with the corresponding tRNA anticodon that carries a specific amino acid is ensured. Let's watch this elongation process of translation in action. Termination of translation begins when a stop codon is in the A site of the ribosome. Stop codons include the sequences UAA, UAG, or UGA. These 3 sequences are special because no tRNAs have anticodons for these sequences. Upon aligning with the A site, the stop codon is recognized by protein release factors that resemble tRNAs. The release factor instructs peptidyl transferase to add a water molecule to the carboxyl end of the P site amino acid. This reaction forces the P site amino acid to then detach from its tRNA, and the newly made protein, or polypeptide, is released. The small and large ribosomal subunits then dissociate from the mRNA and from each other. They are then almost immediately recruited into another translation initiation complex, ready to take on the next strand of mRNA that leaves the nucleus. After many ribosomes have completed translation, the mRNA is degraded so the nucleotides can be reused in another transcription reaction. So now we have our polypeptide. This means that our forming protein now has all of its amino acids, but its journey is not over yet. Recall back to Unit 0 material. The shape of a protein is very, very important to its function. So now there is a polypeptide in the cytosol, or rough endoplasmic reticulum of the cell, and this new polypeptide must fold into a distinct 3D shape in order to function properly. Some polypeptides go on one step further and go on to join with other polypeptides to make a multi-part protein. Now, many proteins are actually decently good at folding on their own, depending on the environment that they're in, but some polypeptides need some help with proper folding. So, in the cell, we have helper molecules that assist in protein folding, and we call these molecules chaperones. Some proteins also contain special amino acid sequences that direct them to certain parts of the cell. These sequences, often found close to the ends of the polypeptide, can be thought of as the protein's quote-unquote train ticket to its final destination in the cell. We'll talk more on this process in our next discussion. But for now, tying this all back together with what we discussed earlier, we have our T cell. In the nucleus of that T cell, we had DNA. Within that DNA, we had a gene that had directions for making proteins, specifically cytokine proteins. And so we transcribed that one gene into mRNA so the mRNA could leave the nucleus, where it was taken up by a ribosome, so the ribosome could then translate that code into a functional protein, specifically our cytokine proteins. And now there is assisted immune responses within this cell. that can help out with the larger organism that the cell is contributing to. Makes sense? And this process is also the same that would be followed when you're looking at why somebody's eyes are darker than somebody else's, right? The genes in the DNA code for proteins, and then those proteins can contribute to things like eye color. And so while in this example we used a protein that has a specific function, some functions of proteins are related directly to phenotype. I hope you found this helpful, and we'll talk more about how we transfer proteins all around the cell moving forward.